And now I would like to welcome Professor Dr. Andreas Herrmann. Enjoy the film.
Hello everybody, welcome to my presentation. I hope I can give you an inspiration today of what you can do with highly charged polypeptides that are generated by recombinant methods. Before we go to supercharged polypeptides, I would like to start with folded proteins and the supercharged variants. In the middle left, you see green fluorescent protein as the native form, and what Mother Nature with most of the protein is doing is it puts negatively and positively charged amino acids on the surface to stabilize them in solution. With genetic engineering methods, however, it's possible to change the positively charged amino acids into negative ones. Then you obtain highly negatively charged variants like the -30 GFP, or you can do the opposite and then you get super positive variants of GFP like shown on the right hand side. We are, however, dealing not with folded proteins, we are dealing with non-folded proteins, and as an example for that, we have chosen for elastin-like polypeptides. These are repetitive structures that are present in our blood vessels and are responsible for the elastic properties. These materials usually consist of a pentapeptide repeat motif that is shown there, VPGXG. Mother Nature puts at the position X usually a valine, so it's a neutral protein. However, this fourth position opens the opportunity to introduce charges. When we put at position X a lysine, then we get positively charged variants. If we put a glutamic acid, then we obtain the corresponding negatively charged variants. How do we produce the structures? We encode the peptide sequences that we want to have in the final polymer structure. We encode them on the genetic level. That means we construct a piece of DNA that encodes our sequence. This we put in a circular DNA plasmid. The plasmid we transform into bacteria, and then we tell the bacteria to produce the proteins for us. So the bacteria are the synthetic chemists in the laboratory. Finally, we just lyse the cells and we purify the protein, and then we can use it for different applications and we can characterize our material. On this slide, I would like to give you an impression of what structures are accessible. We can make structures that contain from 18 to 144 charges. For the negative variants, we can make them charged less, so less than every 5 amino acids, then every 10th amino acid is charged, or we can increase the charge density along the polymer chain, and then almost every second amino acid carries a charge. For the positive variants, we can make similar structures. However, the maximum here is that every 5th amino acid is charged. If you look at the lower panel, you can clearly see that we can use these unfolded structures and fuse them to any other folded protein to make this type of fusions, as shown here for the green fluorescent protein variants. It needs to be pointed out that here that the structures that we do are very, very precise. They have no dispersity like synthetic polymers. We have perfect control about the position of the charges along the chain, and we have a perfect control about the number of charges that are present within one chain. And this is in stark contrast to synthetic polymers, where it's very difficult to place charges equidistantly and to perfectly control the number. In the following few slides, I would like to show you now what we can do with the pristine supercharged polypeptide materials. Then I will show some examples where we complex our structures with other charged biomacromolecules. And finally, I would like to show you what happens once we combine these supercharged polypeptides with surfactant molecules. Let's start with supercharged polypeptides on a surface. Here we have placed differently charged, so positive or negatively charged variants on a surface, and then we investigated how this polypeptide coating influences the freezing temperature of water on the surface. And what you can see in the channel on the right hand side at the bottom, you see that by varying the nature of charge and varying the charge density, we have very good control over the freezing temperature of water on a surface. And that can be, for example, very interesting for biocompatible anti-icing coating. In the next example, I would like to show you that we can remote control our protein structure with the help of this unfolded structure. And that brings us first to the motivation. And the motivation for doing this is optogenetics. Optogenetics had tremendous impact on the life sciences and also on medicine because it allows to remote control biological systems by light. However, if you take a look at the red that is shown in the middle, this is hampered by severe drawbacks of light. Light can not penetrate deep into tissue, and that means for all these optogenetic experiments, one really needs surgical interventions, as you can see from the fiber that goes through the skull into the brain of these animals. We are proposing a different strategy. Our aim is to control protein function by ultrasound. Why ultrasound? Because it has a very deep penetration depth and it is employed in the clinic for more than 5 decades. Here's now a first example how we turn on the activity of a protein. In this case, we have chosen for green fluorescent protein, which is easy to monitor. In this case, we engineered a loop structure, as you can see on the left-hand side, into the barrel structure. And this loop structure is a biophysical transducer for ultrasound. Once you apply ultrasound to a solution, you generate cavitation bubbles. And once these cavitation bubbles implode, they transfer shear forces on this unfolded polymer chain. And this pulls out the 1 beta sheet out of the 10 beta sheets that remain into the structure, as can be seen on the right. And by this, you switch off the fluorescent properties of this protein. Here are some experimental data that really document that this works very well. The black line in the curve shows you here we have used green fluorescent protein structure with a very small loop, so there's no chance of transferring shear forces on this protein. This means upon ultrasonication, there's no influence on the protein function. However, if we now engineer into the structure a loop with 36 negative charges, then within 10 minutes of ultrasonication, we almost switch off the 50% of the fluorescence activity. If we make this loop structure larger, then the protein gets more sensitive towards ultrasound, and then within 4 minutes, we can completely switch off the fluorescent properties of our protein. It's also nice to turn on the activity of a protein. Therefore, we have chosen for a different design. We start with protease. In this case, it's trypsin. That's the green structure. And into the active site of this green structure, we put the trypsin inhibitor that's shown here in blue. And again, the blue structure is modified with an unfolded polypeptide chain that is now positively charged, which again allows to transfer shear forces. And by ultrasonication, we now pull out this inhibitor out of the active site. And by this, we switch on the proteolytic activity of this enzyme, as can be seen on the scheme on the right. The black curve is the trypsin in the active state without inhibitor. The curves on the lower edge of the graph, they show the switched off protein, and once we then only sonicate for 30 or 60 seconds, we regain significantly enzyme activity. Let's move now from the pristine submaterials to materials where we do combinations, and especially we exploit here charge-charge interactions to manipulate and achieve functionality. I would like to start with an alternative treatment modality for Sjogren's syndrome. Sjogren's syndrome is a systemic autoimmune disease. In this case, the immune system attacks cells in the oral cavity that are responsible for producing saliva. From this disease, quite a large fraction of the population are affected, and the symptoms are usually a dry eye and a dry mouth, and patients have a lot of problems with speaking, with mastication, and with swallowing food. Why do the patients have these symptoms? The answer is because mucins are present in the saliva and there's too little saliva produced. So the lack of the mucins leads to the dry mouth effect. The reason is once the mucins are present, then they can, with negatively charged sugar units, they can attract water molecules as shown on the bottom. And then once these mucins are placed on the surface, then articulating surfaces can easily glide against each other with very low friction. The current treatment is that patients usually get a spray of negatively charged polymers or mucins that are recovered from cows and pigs. Our treatment takes the opposite way. Instead of treating with negatively charged polymers, our aim is to use the mucin layer that is still present, not to the same extent as in healthy patients, however, it's present. And these mucins we want to stabilize with the supercharged polypeptides, and we engineer the supercharged peptides with such a chain length that there is always excess charge present on the surface, and with this excess charge we recruit new mucins that are produced, and by this we produce a very stable and smooth layer that allows low friction. Here are the experiments that prove my statement from before. On the left-hand side, you see the colloid probe AFM measurements where we have placed in collaboration with Henk Bücher and Professor Sharma. We have put a mucin layer on the surface, and then we measure the friction forces on this surface, and from the graph on the left-hand side we can conclude, or we can calculate the coefficient of friction of human saliva, which is around 0.2. However, if we do the electrostatic architecture that I've shown you on the slide before, so mucin layer, supercharged polypeptide, and additional mucin layer, we do the same measurement, and from this we calculate the coefficient of friction that is significantly lower and even lower than the friction coefficient of native saliva. In the next step, we have taken this further, and now we have not taken saliva from healthy individuals, but we have taken it from Sjogren's syndrome patients. We have now also gone away from microscopic experiment to a macroscopic ex vitro model experiment that's shown on the bottom right, where we take enamel on one surface and a cow tongue on the second surface, and then we do macroscopic friction experiments. What we learn from these friction experiments is once you just add the patient saliva, the existence of this layer is not durable for a long time. That means after a couple of minutes, around 3 to 4 minutes, the low friction phase is lost, and then patients would again need to spray the new saliva. However, if we stabilize our protein or mucin structure with the protein, then we obtain a very stable layer that holds for more than 20 minutes, and this leads to great relief for the patient. Let's move now from a therapeutic application to a sensing application. In this case, now we use green fluorescent proteins that are fused with supercharged polypeptides. This is a collaboration with Professor Uwe Bunz in Heidelberg. In this case, we construct a chemical tongue, but let's first have a look at what a normal tongue in our mouse is doing. We, as humans, we are able to distinguish between 5 different tasters that are shown here, and with different intensities of these tasters, we get certain taste sensations. So this more or less works like a matrix, and something similar we make with the chemical tongue. So we take the fluorescent proteins and we put it in individual wells of a multi-well plate, and we use conjugated polymers, and then for each of the analyze we add to each of the individual component, we get either a fluorescence quenching or a fluorescence increase, depending how the analyte interacts with the sensor matrix. In this case, we used fluorescent proteins from our lab, and we used conjugated polymers from the Uwe Bunz lab. We placed them in these arrays, and then we added 50 different whiskies, and we were able to distinguish these whiskies by taste, brand, age, and origin. What is usually does, we, after measuring the fluorescent signals, we need to do a mathematical analysis, and then we can plot our measurements in these 2-dimensional plots. Also, it is a multi-dimensional matrix, and then we get really clustering of the results, which shows that we can clearly distinguish between different whiskies as shown before. And I just want to refer to the Bowmore, which is one whiskey that I really like myself. And here you can clearly see that we can distinguish the 12-year-old from the 15-year-old from the 18-year-old. So let's move now to the last part of my talk, where I would like now to show you electrostatic complexes between supercharged polypeptides and surfactants. This brings me first to the field of liquid crystals. And if you look at the liquid crystal molecule at the top, you can see that these molecules, they usually consist of a rigid part and a flexible part. And then there are 2 major thrusts of liquid crystals one needs to distinguish. These are on the first hand lyotropic liquid crystals. In this case, the mesogens, as shown in the top, they are dissolved in a solution, and depending on the concentration, they take different liquid crystalline structures as seen on the right-hand side. And then there exist thermotropic liquid crystals. In this case, the mesogens, as shown on the top, are forming a bulk material. In this case now, we can adjust the liquid crystalline properties by varying the temperature. For biomacromolecules, there have been lyotropic liquid crystals reported, like for these short DNA strands that are shown here on the slide. However, there were no thermotropic liquid crystals reported. Also, these are the more important ones if you consider, for example, liquid crystals in displays, which allowed the portable computer revolution. The fabrication of this protein thermotropic liquid crystals is pretty simple. You just take the supercharged polypeptide and you complex it with a cationically charged surfactant. Once you combine both in solution, you get a precipitate. You remove the aqueous supernatant, and then you can immediately characterize the thermotropic phases. On the top left, you see that we obtain a very stable liquid crystalline phase between 31 and 75 degrees C. On the right-hand side, on the top, you see a typical focal conic texture under polarization optical microscopy. And from Sachs and Wax measurement, you can also calculate the structural properties of these materials. And from this, we know that we obtain smectic structures, so layered structures with surfactant double layer that is separated from the next layer by the supercharged polypeptides. If we now reverse the nature of charge, we now take a cationic supercharged polypeptide and we complex it with a anionic surfactant that contains an aromatic head group. Then we get a precipitate if we add both components together in an aqueous solution. However, in this case, we don't get a hydrophobic precipitate without water. In this case, we get a coacervate that still contains a bit of water. And again, once we use this coacervate and we put it on the surface, then we observe very intriguing adhesive properties. This we have characterized, and we have characterized these adhesive properties on different surfaces, and we have compared it to commercial adhesive, which is cyanoacrylate. In colloquial language, this is called superglue. And what you can easily see is that with the genetically engineered glue, we obtain very similar adhesive properties as with the superglue. We did some structure property relationships, and we found out that the longer the supercharged polypeptide chain is, the better the adhesive properties. Moreover, we did very elaborative studies what contributes to the cohesion forces into the glue structure, and we found out that the adhesive and cohesive forces are dominated by electrostatic and cation pi interactions. And due to the fact that we deal here with a protein structure that's biodegradable and we deal with a surfactant that's approved for cosmetics, we can use these glues in vivo. Here, in collaboration with Professor Kai Liu at Beijing University, we installed cuts at the back of a mouse, and then we looked at the healing properties. We have compared 4 different experiments. First, a wound that was not treated, that's in the first row. Then we examined the wound that was closed by suture. The third row shows treatment with a commercial medical glue, and the fourth row below shows the treatment with our genetically engineered superglue. What you can clearly see is that our material does not only have adhesive properties, but that it also strongly promotes the wound closure and wound healing as documented in this animal experiment. With this, I'm at the end of my presentation. I would like to conclude with a short summary. I've shown you that supercharged polypeptides can be preferred as perfectly defined polyelectrolytes. Complexation with other molecules by electrostatic interactions allows new structures and functionalities. I've shown you that fusion with other protein fragments leads to multifunctional hybrid materials. And what is, in my opinion, very striking is that also we have no folded structures. We can release very different functionalities in solution, on surfaces, and in the bulk. Finally, I would now like to thank the people that contributed to this work. I'm only the person presenting, but a great thanks goes to my co-workers, PhD students, and postdocs. They are responsible for these results. The same holds true for my collaboration partners. Many of them I've already mentioned along in the talk. I would also like to thank the organizations that gave me the money to do this research. And finally, I would like to thank you for your attention, and I'm now looking really forward to a discussion with you.
Thank you very much for the introductory presentation. Professor Dr. Herrmann will now join us live. Welcome. We understand you are at a conference in Bordeaux right now. How nice.
Yes.
So, how's the weather?
Morning, everybody. The weather is fine. It's almost 25 degrees already, and it will get warmer today. But I heard that in Germany the weather is also being good. So I think we all enjoy, and I hope you enjoyed my talk. And now I'm really looking forward to questions.
Yes, it is indeed good, and we are also looking forward to having you here. So thank you for this. And we of course have already interested viewers who have some questions. Peter, it's your turn. You can ask the first question.
We got the first question, and thank you very much for the nice talk. This is also interesting for me as a compounding company, compounder. And the question is, are these polypeptides thermally stable for processing with bioplastics, e.g. as a compatibilizer?
Yes, the thermal stability of these materials is pretty high. So you can think of that they are as stable as, for example, nylon. So to use them in compounding applications is, in my opinion, feasible. Also, we have not yet tried this out.
Yeah, I hope we will change that soon. So we should stay in contact.
Okay, so we have another question coming in from the chat. How about the biodegradability of the polypeptides and the productivity of the biosynthesis?
Yeah, that are two good questions regarding the biocompatibility. We have, for example, investigators in the wound healing experiments where we have used as a glue the variant that contains of a fusion of the anionic sub and the GFP, and then we saw that in the wound, within 1 to 2 days, the material was almost degraded completely. We have done in vitro experiments with proteases, so the degradability of these materials is very, very good, depending on the place where you use it in the human body. And regarding the second question that regards is basically the question how much and how efficient you can produce these materials. That depends a bit on the chain length that you want to pursue, the nature of charge that you want to introduce, either the positively charged or the negatively charged ones. So far, we use E. coli as the expression host, and the efficiencies, they vary between, let's say, 20, 30 milligrams per liter cell culture on the lower side to up to 200 milligram per liter on the higher efficiently producible side.
Okay, then there comes a question that is, you already mentioned something like that. What molecular weights can be realized?
The molecular weights that we can realize, the largest variant that we have done is contains 144 charges, and this then leads to chains that are around 600 to 700 amino acids long, and the molecular weights that result from those are in the range of 70 to 80,000 Dalton.
Okay.
So what can produce high molecular weight materials with these methods?
Okay, yeah, that's an interesting project indeed. We still have some time, so we have lots of questions coming in, which is good. So the next question is, how fast does your super glue cure?
For all the experiments we have done, we have put the material on top of the surfaces. Then we have clamped it together with a paper clip for 1 day, and then we have performed the measurements and determined the adhesion strength. Another experiment that we have done for cosmetic applications is we have put the so in Asia, the females, they want to have a eyelid, artificial eyelid, which many of them don't have, and for that purpose we have put the subglue on the eyelid, and then we put in this and then you induce the formation of this feature, and in this case the material glues together the skin within less than a minute, and you apply it, and instantly you have the effect, the cosmetic effect that you wish.
Wow,
Okay.
that's very impressive. Okay.
Then there's
Yeah, so this is described in the Nature Communication papers. There you see the cosmetic application. There this is described in more detail, and there are also some photographs showing how the glue works and how it can replace a double-sided tape application that is used in the market.
Okay. Then maybe the last question, how can it be applied to the surface?
The application, depending on what application you do, for example, for the glue, you have the material still contains a bit of water, and the viscosity is not so high. I would say that the viscosity is like a commercial glue, like for example Uhu or Patex. So it has this a honey-like viscosity, and then you can apply it with a spatula on a surface, and then you can apply the second substrate to produce the adhesive joint. For example, the anti-icing applications, there you just apply the material in an aqueous solution, and due to electrostatics, the material sticks to the surface, and with this you can induce the different icing temperatures on these surfaces. So it depends on the application, but you can apply it from solution, or you can apply it as a coacervate, which has a handleable viscosity.
Okay, that's very interesting. Maybe we can use it next year for our trophy, for the awards trophy, because there are the sheets glued together. So we could use also the biological glue.
Perfect idea, Peter. That's it.
At least this would be a contribution to the sustainability of your conference. I very much support your suggestion.
Okay.
So we have a cooperation for the next year. Might have a cooperation, right?
It is biological
Very
plastic
good.
and in combination with biological glue. Yeah, that's good.
That's really good. Thank you very much, Professor Dr. Herrmann, for being with us, for answering the questions. It was very interesting. We're wishing you a great time in Bordeaux. Enjoy the French wine and have a good time. Thank you.
Thank you
for
very
being
much.
here.
Yeah, thank you very much for inviting me to show some of our results, and I wish you all a very fruitful and interesting conference and a very good time. Thank you very much.
Thank you.
Thank you very
Bye-bye.
much. Bye.
Bye-bye.